Now, one important feature of these global control systems is that they don't necessarily affect every mRNA in the same manner. And I want to illustrate that by using a simple mathematical model for the process of translation initiation. And the larger point I want to make here is that the global control of translation affects mRNAs differently due to their different rate-limiting steps in initiation. So let's consider the process of translation in a very simplified view of having three discrete steps. In the first step, we would assemble this cap binding complex on the mRNA. In the second step, the small ribosomal subunit would scan down the message, find the AUG codon. And in the third step, the large ribosomal subunit would come in and allow for protein production to begin. Now, we can mathematically model this, and in doing so, we're going to assume that this third step, the joining of 6DS subunit, is relatively fast compared to the other two. And there's evidence in vivo that suggests that's generally true. All right, so what happens if we do that? And then we look at the amount of protein production for how those rates change. And that's illustrated in this slide, where this is basically a graph showing protein production as a function of the rate of assembly of the translation complex and the rate of scanning and AUG recognition. All right, so let's consider two different mRNAs. So if we look down here, this axis here is the rate of assembly. So consider an mRNA whose assembly is slow. Okay, so this would be right here. If we change the rate of scanning and AUG recognition from fast to slow, it doesn't really change the amount of protein production, right? And that's simply because the rate limiting step is the rate of assembly of the initial translation complex. Conversely, consider an mRNA where the rate of assembly is fast. Okay, so that would be out here. Now, if we change the rate of scanning and AUG recognition from fast to slow, we get a significant change in the amount of protein produced. When the rate of scanning and AUG recognition is fast, there's a lot of protein. Now, if you decrease it, there's less protein. Whereas for this RNA, there's relatively little change at all. Okay? So the important point here is because you have different rate-limiting steps, changing a given step may affect an mRNA the same or different from other mRNAs. And I want to emphasize this is a general principle of any pathway which has multiple steps where different ones can be rate-limiting for different substrates. And this is a reoccurring theme both in translation and also the steps of mRNA degradation, as we'll discuss in a few minutes.